Now, you probably remember learning about the cycle of cell division in grades 8 and 9, but if not, for whatever reason, here's another run-through. The cell cycle occurs in 4 stages: G1, S, G2 and M. In G1, the G stands for growth, the organelles of the cell double in number. In S, synthesis, DNA is replicated, ready to be divided into new cells. In G2, various proteins are synthesized. These proteins will activate the next stage of cell division. The fourth and final stage of the cell cycle, M, mitosis. The amount of time all this happens in is called interphase. Got it? No? Too bad. Wash and break? Course not. Science is happening. Cell division occurs in a couple of ways. The first is called mitosis. It is the nuclear division that produces 2 daughter nuclei, each with the same number of chromosomes as the parental nucleus. Get it? No? Well, it looks like this. Mitosis happens in 5 stages. The first is prophase. The centrosomes of the cell begin to move apart from one another, and the nuclear envelope disintegrates. Inside are the chromosomes, which attach onto the centrosome's spindle fibres. Now the second stage, metaphase. Here, the chromosomes line up along the cell's equator, or the metaphase plate. Easy to remember. Stage 3 is anaphase, when the chromosomes are pulled apart by the chromosome spindles to either end of the cell. Now, both sides have an equal number of daughter chromosomes. Fourth is telophase. With daughter chromosomes at either end of the cell, a furrow appears between the 2 groups and begins to cut them apart. Meanwhile, a new nuclear envelope is assembling around the chromosomes. Asleep yet? One more stage, cytokinesis, which, quite simply, splits the cell into 2 new ones. Ta-da! It's a beautiful set of twins. Remember those proteins I mentioned earlier? The ones synthesized during interphase? Well, these proteins regulate the cell cycle. When activated, they trigger each step of the process. They are comprised of 2 enzymes, cyclin and kinase. Cyclin is an enzyme that attaches to kinase to activate it. Kinase is an enzyme that steals a phosphate group from an ATP and gives it to a protein. This protein then activates the next stage of the cell cycle. Easy. It all works like this. In G1, the kinase connects with cyclin and takes a phosphate group to give to a protein. This triggers synthesis, stage S. Then the whole thing breaks apart and disintegrates. In G2, the same thing happens. Kinase gets with the cyclin, a phosphate group is taken, and the protein triggers mitosis. If you like, the G stages are the dressing rooms, where all the enzymes get ready to perform synthesis and mitosis. But how do kinase and cyclin know how and when to do these jobs? The answer lies in growth factors, the policemen of cell division. They are, simply put, signalers that direct each enzyme, protein and molecule through the process of cell division. But, as you know, there is always a rebel. When enzymes don't follow the given signals or carry on without a growth factor present, they go into overdrive and, with no way of stopping, mass-produce daughter cells. This is how cancerous tumours are formed. Now to wrap up, I'm counting 3 sleepers. Need a quick review? The cell cycle is in 4 stages, G1, S, G2 and M. The cell division, mitosis, is in 5 stages, prophase, anaphase, metaphase, telophase and cytokinesis. Mitosis is driven by the 2 enzymes cyclin and kinase. These are controlled by growth factors. Lack of growth factors means overproduction of cells and forming cancerous tumours. And that's it! You're free! Go home! Thanks for watching.